Hi there. I just wanted to give you a quick recap of the mini lesson that we covered last Monday, January 4th, during our first meeting together. And I'm here on the modules link in Canvas, and hopefully you all successfully completed the items under Start Here, Orientation and Syllabus. I'll be grading those over the next few days. And just a reminder, some of these are just files that you review, some of them are attachments, and some of those are actually wiki or HTML pages that I created in Canvas. And then some of these, anything that shows that it's worth points is an actual assignment. So that was the orientation syllabus. And last week you did week 1, and hopefully you started by looking at the week 1 to-do list, which was a page that reminded you, hey, you're new to the Canvas site, so keep looking at it. Get familiar with the textbook. And I had the orientation items keeping Unit 1 locked, but I actually unlocked for those for you so that you could access week 1 once I saw that you were completing orientation assignments. Then you started the Chapter 1 assignments in Lab Module 1, but those weren't due until Saturday. And then all of this listed here was due on Saturday night at midnight. So let me go back to modules. And then here, wait a second for it to collapse. And that was the week 1 to-do list. Now what you want to do for week 1 is hopefully you've opened the PowerPoint and walked through and spent some time with it. And I'm hoping that you took some time to open this page where I have curated videos that help you become more familiar with the scientific method as a process, with the characteristics and the classification of life, and then more about studying biology and classification. All great videos for you to watch and some nice reinforcing material there. Okay, let's go back to modules. And then you can see for week 1, you had a couple of learning assignments, so guided studies to help you become more familiar with the material for Chapter 1, and a coronavirus reflection to keep us current with the times instead of a project. And then the Chapter 1 study guide, I'm going to open for a minute. And the first 2 study assignments, you're using Cornell Note style, and there were a couple of videos in case you didn't know what it was, and some examples. And remember, hopefully, to write your summary at the end. And here's how to proceed, even when you don't take Cornell notes anymore, if you don't like them and you're using another technique for note taking, is that you would follow this process when you study. Read the content objectives without expecting to know the answers, just to get a feel for important information. What's this week about? If you can print them, do that, or copy and paste them into an electronic file so that they're next to you when you refer to other material. Read the textbook sections and try to process what's going on in each subsection. Basically, you're trying to make sense of it and get familiar with the ideas without having to memorize everything. Then, now that you've looked at the content objectives in the textbook, go through the PowerPoint and think, what are the things that are being emphasized in the PowerPoint that I saw in the textbook that are emphasized in the content objectives? What is it that I need to know? Look at the weekly objectives, draw a connection. Then work on the learning assignments relevant to the chapter and section of the chapters that are due that week. And you may work on one section of a chapter and do this process and then start the next section. So then you go back through the learning objectives. What do you think you could explain at this point? What makes sense? Start making a study guide, whether it's the Cornell note style or some other type of mapping or note style that you do later. Leave some space where you can't answer or you don't have enough information. Use the objectives to organize and direct your notes. In Cornell note style, you would use those to write reflective analysis level thinking questions on the left side of the note-taking sheet. Then watch some videos while referring to the content objectives. Take some notes. Focus on the things you're struggling with and try to fill those concepts in the study guide. See how the study guide is really a tool for you and not something that I'm going to read or use. It's me looking to see, have you been using the content objectives? Are you learning how to layer your learning? And then basically you can see how you can get to 12 to 15 hours a week, because it's not that writing the notes takes you that long, it's that this whole process gets you through all of the other assignments related to learning the material, and you submit your study guide and you submit your learning assignments and you complete the lab and you complete the coronavirus reflection and you turn all those in by Saturday night. So here we go, back to modules. And I want to then say here down at the last bit of every week is what is on the lab. And I walked you through the lab with last Monday night, and I also have quite a bit on our Zoom recordings from the spring, so that would be in here with Zoom sessions. And this week 1 help, you can see scientific method, help with learning assignment number 2, help with lab module 1A, help with lab module 1, a little bit of Canvas and how to use it mixed in there. And what I'm going to do now is go to the PowerPoint and say, hey, when you go back and you review the PowerPoint, these are the things that I want you to focus on. So I'm just zooming in a little, maybe I'll zoom out a little bit. So here I have it with 4 slides in a row, and I want you to just focus on what are the key things that are happening in this PowerPoint as you go through. What is the history of science and with the way people think and the way they reason? You know, what's the difference between something that's concrete knowledge versus abstract knowledge? You know, what you feel, what you imagine versus what you can, you know, see and what you can touch, what you can measure. And when we think about modern science, it's this body of practical knowledge and theoretical knowledge, but it's based on evidence, data gathered from observation and analysis of the natural world. So when we want to make accurate observations, we have to do it without bias, and we have to use a process that's going to eliminate bias. And that is the goal of the scientific method. I want you to focus on inductive reasoning and deductive reasoning as 2 ways of using and applying the scientific method, where inductive reasoning draws conclusions here through the process of observation. So lots of singular individual observations lead to a collective body of knowledge that is a generalization that would help you develop a scientific theory, a big explanation for how a particular process works. And then if you got a piece of new information and you wanted to figure out how to fit it into your theory and you needed to test it, well, when you're testing a little specific example and to see if it fits into your broader idea, that is deductive reasoning. So hypothesis testing is deductive reasoning, theory development is inductive. So when we were thinking about cells and how living things are organized and what's the smallest thing that can be alive, and the microscope was invented, thousands and thousands of observations with the microscope led a group of scientists to come up with cell theory, which we'll cover in short order here in the coming chapters. And then when we find something new and we look it up under the microscope, we can decide if that little microscopic thing is alive and is made of cells or if it's not. And so we used inductive reasoning to make millions of observations to come up with cell theory, and then we take something new that we want to see if it's a cell and apply hypothesis testing and deductive reasoning to determine if that hypothesis is true. Okay, so coming back out to the full PowerPoint, the next dozen or so slides go through hypothesis and theory and the steps in the process of the scientific method. And in my online lesson, I talked about this slide, the scientific method applying the process, and about the steps and you know, making observations. And then if you heard my daughter ask where the vacuum is, then you know it's not relevant information. Okay, sorry about that interruption. You probably heard the phone ring. It was my brother in New York who wanted to update me about his COVID test. Okay, so after you go through the steps of the scientific method and you watch a couple of videos, hopefully you took some time to work your way through these practices. If you haven't done that, you could still work on one and, you know, send me a picture and see if you got it right. Then the next thing to focus on is these 7 characteristics of living things. Hopefully learning assignment number 1 really helped you become comfortable and confident. If you still have any questions about that, you can email me. Again, there's also a great video about these. And one of the things to know when you're tested is it's not just can you memorize this list, but can you explain what it means to respond to stimulus and give an example. For you know, if I touch a special kind of leaf on a plant that exhibits thigmotropism, then when the response to the touch stimulus is that the leaves close. Sea anemones do the same thing. And so responding to light, to heat, to touch, to an electrical impulse, when cells and a eukaryotic multicellular organism responds to something from the outside, that's response to stimulus. So just be able to explain it and give an example. And the next many slides do that. And then I want you to take some time and see if you can understand the characteristics of life that are illustrated. So, you know, shot up 5 inches in 1 year, that's growth. Having a litter of kittens, that's reproduction. Becoming much less clumsy, that's development. Eat a good breakfast and you will be able to run longer. Well, what are you doing with the food? You are using energy, right? So just work your way through those. Then the next couple of slides talk about organization and about how living things are organized into the domains. So take some time and go through this. We don't spend lots of time on taxonomy in Biology 160, but it is important to understand the 3 domains, especially when we get into chapter 3 and cell theory and the way things are organized. And I also want you to understand that living things are organized based on, you know, the smallest things that are contained within cells, then the way cells are organized into tissues and organs, and that those make organisms, and that organisms themselves don't live alone, but they live in populations and they live with groups of other organisms and communities. They interact with the abiotic or non-living environment in ecosystems and, you know, at the community level, and that all of that together on the planet makes the biosphere. You got a chance to practice a little bit of this at the very end of learning assignment number 1, and these slides go through that and you get a little practice. This was on the learning assignment. And if you're not quite sure what the answers were, you could check in with me. So that was PowerPoint 1, and that's the end of my mini lesson.